Every time your body feels threatened, whether it's a looming deadline, a sleepless night, or chronic inflammation, your adrenal glands release a powerful hormone, cortisol. In the short term, cortisol is your survival engine. It mobilizes energy. It sharpens focus. It keeps you alive under pressure. But here's the problem. When cortisol stays elevated, day after day, week after week, it quietly hijacks your blood sugar system. And for millions of people, that hijacking becomes type 2 diabetes. Today, we're going deep. We're tracing the exact molecular pathway by which cortisol drives glucose into your bloodstream, overrides insulin's signal, and eventually breaks down the system designed to protect you. This is glucocorticoid signaling, and once you understand it, you'll never look at stress the same way again. Cortisol is a glucocorticoid, a steroid hormone produced in the adrenal cortex, the outer layer of the adrenal glands that sit just above your kidneys. Its release is controlled by the hypothalamic pituitary adrenal axis, or the HPA axis, one of the most important regulatory circuits in your body. Here's how it works. The hypothalamus in the brain detects a stressor and releases corticotropin-releasing hormone, or CRH. That signal travels to the anterior pituitary, which responds by secreting ACTH, adrenocorticotropic hormone. ACTH then reaches the adrenal cortex, triggering the synthesis and release of cortisol. Under normal circumstances, rising cortisol feeds back to the hypothalamus and pituitary, shutting the signal off. This is called negative feedback, and it's what keeps your cortisol levels in a healthy rhythm. But in chronic stress states, whether psychological, inflammatory, or metabolic, this feedback loop weakens. Cortisol stays high, and that's when the real damage begins. Unlike most hormones that work by binding receptors on the cell surface, cortisol is a steroid. It's lipid-soluble, which means it can cross directly through the cell membrane. Once inside the cell, cortisol binds to the glucocorticoid receptor, or GR, a protein that normally sits in the cytoplasm, waiting. When cortisol binds, everything changes. The GR undergoes a conformational shift. It releases its chaperone proteins, including HSP90, or heat shock protein 90, and the entire cortisol-GR complex translocates into the nucleus. Inside the nucleus, the complex binds to specific sequences on DNA called glucocorticoid response elements, or GREs. These GREs are promoter regions that control the expression of target genes, including the genes responsible for producing glucose in the liver. This is called genomic signaling, and it is the foundation of cortisol's metabolic power. The liver is the body's primary glucose factory, and cortisol has a direct key to the factory floor. Once the cortisol-GR complex binds to GREs in hepatocytes, that is, liver cells, it dramatically upregulates two critical enzymes, phosphoenolpyruvate carboxykinase, or PEPCK, the rate-limiting enzyme of gluconeogenesis, the process of making new glucose from non-sugar substrates like amino acids and lactate, and glucose-6-phosphatase, or G6-PASE, the enzyme responsible for the final step of releasing free glucose into the bloodstream. Together, these two enzymes turn the liver into a high-output glucose producer, even when you haven't eaten. Now here's the critical conflict. Insulin does the opposite. Insulin signals the liver to suppress gluconeogenesis. It activates a pathway through PI3K right-pointing arrow AKT right-pointing arrow FOXO1, which silences PEPCK and G6-PASE expression. But when cortisol is chronically elevated, it directly upregulates FOXO1 transcription, effectively overriding insulin's suppressive signal. This is the core molecular conflict in glucocorticoid-driven hyperglycemia. Cortisol and insulin are pulling liver gene expression in opposite directions, and with chronic cortisol elevation, cortisol wins. Liver glucose overproduction is only one part of the story. The other side is what happens in your peripheral tissues, your skeletal muscle and adipose fat, when cortisol stays high. Normally, when insulin binds to its receptor on a muscle cell, it triggers a cascade. Insulin receptor activation right-pointing arrow IRS1 phosphorylation right-pointing arrow PI3K activation right-pointing arrow AKT activation right-pointing arrow GLUT4 translocation to the cell surface. GLUT4 is the glucose transporter. When it moves to the membrane, glucose enters the cell. Blood sugar drops. The system works. But chronic cortisol disrupts this cascade in several ways. First, cortisol induces IRS1 serine phosphorylation, an inhibitory signal that prevents proper IRS1 activation, effectively jamming the first relay in insulin's chain of command. Second, cortisol upregulates P10, a phosphatase that degrades the PI3K product PIP3, reducing AKT activation. Third, cortisol suppresses GLUT4 gene expression itself, reducing the total pool of glucose transporters available for translocation. The result? Muscle cells become deaf to insulin's message. Even when insulin is present, even when the pancreas is working overtime, glucose stays in the bloodstream. This is peripheral insulin resistance. And in adipose tissue, the situation is compounded by a process called lipolysis. Cortisol stimulates fat breakdown, releasing free fatty acids into circulation, which further impair insulin signaling in both muscle and liver through a mechanism involving ceramide synthesis and PKC theta activation. So we have the liver overproducing glucose. We have muscle and fat tissue refusing to absorb it. What happens next? The pancreas senses the rising blood glucose and compensates. It produces more insulin. The beta cells work harder and harder, trying to overcome the resistance. This phase is called compensatory hyperinsulinemia, elevated insulin levels that temporarily keep blood sugar in range, but at enormous cost to the beta cells themselves. Here's where chronic cortisol elevation becomes catastrophic. Cortisol has been shown to directly suppress beta cell function. Through glucocorticoid receptor activation in pancreatic islets, cortisol downregulates PDX1, a critical transcription factor for beta cell survival and insulin gene expression. It also promotes beta cell apoptosis through BCL2 downregulation and CASPASE3 activation. So the very cells trying to compensate for cortisol-driven insulin resistance are being destroyed by cortisol at the same time. Eventually, the beta cells can no longer compensate. Insulin secretion drops. Blood glucose rises unchecked. The clinical threshold for type 2 diabetes, a fasting glucose above 126 mg per deciliter, or an HbA1c above 6.5%, is crossed. This is the glucocorticoid-driven pathway to metabolic disease. It connects a stress hormone to a pancreatic failure. And it is playing out silently in millions of people right now. The clearest clinical proof of this pathway is Cushing's syndrome, a condition caused by chronically excessive cortisol, whether from a pituitary tumor, adrenal tumor, or long-term steroid medication. More than 80% of Cushing's patients develop glucose intolerance or frank type 2 diabetes. And when cortisol is normalized, through surgery or medication, glycemic control often dramatically improves, even before significant weight loss. This tells us something profound. Cortisol is not just a risk factor for diabetes. In some patients, it may be the primary driver. For clinicians, this has real implications. Patients with poorly controlled type 2 diabetes, particularly those with central obesity, hypertension, and mood disorders, should be evaluated for HPA axis dysregulation. A morning cortisol level, a 24-hour urinary free cortisol, or a low-dose dexamethasone suppression test can reveal subclinical hypercortisolism that might be the root of their metabolic disease. For patients, the implications are equally powerful. Chronic psychological stress, sleep deprivation, and inflammatory conditions all elevate cortisol, and all worsen insulin resistance. High-quality sleep, regular aerobic exercise, and stress reduction are not wellness extras, they are metabolic medicine. Exercise, in particular, increases GLUT4 expression and improves AKT signaling independently of insulin, directly countering cortisol's peripheral effects. Pharmacologically, 11-beta-HSD1 inhibitors, enzymes that convert inactive cortisone to active cortisol in peripheral tissues, represent a promising therapeutic target, with several candidates in clinical trials. The journey from a cortisol molecule to a type 2 diabetes diagnosis isn't random. It follows a precise molecular pathway, from glucocorticoid receptor activation in the liver, to disrupted insulin signaling in muscle and fat, to eventual beta cell exhaustion in the pancreas. Understanding this pathway doesn't just explain the biochemistry. It reframes how we think about metabolic disease, not just as a problem of diet and exercise, but as a product of the entire stress hormone environment that a person lives in, day after day. If you found this useful, subscribe. We go this deep on every topic. And if you want a visual summary of today's signaling pathways, check the pinned comment for a free diagram. Until next time, keep asking why the biology works the way it does. That's it for today. Thanks for watching, and I hope you had a great time. Hit the like button, subscribe, and ring the notification bell so you don't miss future videos. See you in the next video.